So the IMpassion130 trial is a phase 3 trial that randomized women with metastatic triple-negative breast cancer who had recurred at least a year from their adjuvant or neoadjuvant chemotherapy, or who had de novo metastatic breast cancer that was triple-negative, to receive Nab-paclitaxel and placebo, or Nab-paclitaxel and the checkpoint inhibitor Atezolizumab. There were a number of primary endpoints: progression-free survival and overall survival in the intent-to-treat population, and in the subset of patients that had PD-L1 positive disease. And what we found in that study, which has led to approval of Atezolizumab with Nab-paclitaxel both in the US and in the EU, is that progression-free survival was improved in the PD-L1 positive population by about 2.5 months, and overall survival was improved more impressively by 7 months in the final overall survival analysis. And Atezolizumab was relatively well tolerated. The immune toxicities that we've seen with all checkpoint inhibitors weren't any different in that study population. What we discussed here at ESMO actually was data on how we determine PD-L1 positivity, which turns out to be really important because these checkpoint inhibitors are approved for lots of different solid tumors, and different antibodies have been used to determine PD-L1 positivity, and different cut points actually. So it's a bit of a muddy field. So for breast cancer, this is the only approved so far to date checkpoint inhibitor, and the antibody that was used was SP142, and it's the immune cells rather than the tumor cells that are positive. And in additional analyses presented by Leisha Emmons, we found that actually only a small number of tumor cells are positive when the immune cells are not, and that it didn't predict outcome the way the immune cells did. The way you actually score it, it's 1% or more, is the percent area of tumor and stroma where you find those immune cells that are positive by SP142 for PD-L1. In IMpassion130, it was 41%. So what we did was we took a subset of the tumors that were available from IMpassion130 and looked at the other antibodies that are most commonly used to detect SP142. So actually in that subset, 46% were positive by SP142, and the PFS was even a little better than in the parent intent-to-treat trial. But we looked at these two other antibodies, 22C3 and and another antibody that uses a different platform, 22C3, and uses actually a different scoring system called the CPS scoring system, and includes tumor cells as well. And that score 1 or more is positive. And then SP263, which is used for yet another checkpoint inhibitor, a different platform as well, where it's 1% or more. So what we found was that many more tumors were positive for these other two antibodies than for SP142. But SP142 positivity was encompassed within the positive testing for the other two antibodies. And when we looked at double positives and single positives, it looked like SP142 was the most sensitive indicator for the benefit from Atezolizumab added to Nab-paclitaxel. You saw the biggest difference in PFS and OS. But if you did the other antibodies, you would be including that population of patients. You might be over-treating some of the patients. There also has been big questions about where you should get the tumor from. So do you have to biopsy a metastatic site, or could you use primary breast tumor? And we looked at that. Actually, the median time from tumor collection to randomization was only 61 days. So most of the tumor was collected when patients had metastatic disease. But it turned out that PD-L1 positivity was relatively higher in breast primary tumor than in a metastatic tumor site, and that the percent positivity varied a lot based on the tumor site, so that we saw the lowest percent positive in liver, which other studies have shown as well, even though we had a relatively small number of liver biopsies. So this information, I think, is really important and can help direct clinical care, and also will be used, I think, as a comparator when we start looking at other study results. For example, Keynote 522 that will be presented here at ESMO, looking at neoadjuvant pembrolizumab.